Hello, my name is Richard Pither. I'm the CEO of Cytox, and I'd like to talk to you today about the use of polygenic risk scores in the early detection and clinical management of Alzheimer's disease. These are my disclosures, and I would encourage you to read those. Now, the problem we're trying to solve is one of an increasing prevalence of Alzheimer's disease. About 46 million are afflicted today, and that number is expected to double by 2030, largely driven by the aging demographic of populations around the world. Current diagnostic techniques have poor accuracy and tend to be invasive and in many cases high cost. And in none of these techniques are capable of identifying those at highest risk of cognitive decline due to Alzheimer's disease. And of course, that's a major concern for patients, their physicians, and indeed drug companies trying to enrich subjects of that nature into clinical trials to prove the efficacy of their interventions. Our solution to this is GenaScore. GenaScore is a SNP-based genomic test for assessing the risk of developing Alzheimer's disease and the associated cognitive decline. It's easy to perform. We need genomic DNA. You can see from the workflow at the bottom that DNA can come from a mouth swab. It can come from a blood sample. That can be a fresh or an archived blood sample. DNA is extracted and then run on our proprietary array that we call VariATECT. VariATECT is a bespoke Axiom array built for us by Thermo Fisher and read on the GeneTitan platform. And that's read in any lab around the world that can analyze or has that equipment. We work typically with Akesogen in Atlanta, with Eurofins in Europe, and with the Ramachotty Center in Australia. The output from that array contains 500,000 SNP data points for each sample, 96 samples parallel processed, and is taken into our SNPfitter software, which is a cloud-based software which contains the GenaScore polygenic risk score algorithm and can be used to process the data in the location of the sample. So Alzheimer's disease, late onset Alzheimer's disease, has a complex genetic etiology. We'll talk about that in a moment. But of course, that genetic risk is fixed throughout your lifetime. What determines whether that risk becomes manifest at the level of clinical symptoms is a function of exposure to environmental and lifestyle risk factors, of which some are illustrated here. So cardiovascular disease, diet and exercise, diabetes, neuroinflammation, to name but some. So if one knew that one was at relatively high risk of developing Alzheimer's disease by virtue of genetic inheritance, you might take proactive steps to manage risk associated with those factors, thereby pushing out or reducing the rate of decline at later points in life. Now, the late onset form of Alzheimer's disease is complex genetically. If we look here at the Manhattan plot, we can see something like 40 genes that reach genome-wide significance. In fact, if you take the significance level down a bit, we find over 114,000 DNA variants or SNPs that contribute either towards Alzheimer risk or indeed can be protective variants. By far the most dominant of these is shown on the right-hand side of the plot, which is the ApoE gene. And as we'll describe in a moment, ApoE has a major role, but is in no way diagnostic. So you can carry variants of ApoE which give you increased risk, but they don't tell you whether you're going to get the disease or not. A polygenic risk score takes into account ApoE, but it also takes into account all those other loci distributed across the genome. So as an example here, we can see that if we look at ApoE allelic distribution, we can see that the majority of Caucasian subjects, over 60%, carry the E3 homozygote profile, and that's considered to be an average level of risk. But about 25% of the population carry one or more copies of E4, most commonly E4, E3 heterozygotes, and these are considered to be at elevated risk, but in no way would that be diagnostic. Equally important, of course, is that if you are E3 homozygote, it does not preclude you from being high risk for Alzheimer's disease. Indeed, something like 40% of those diagnosed with Alzheimer's disease come from that E3 homozygote group, But of course, there is no way to stratify them just using ApoE4 genetics alone. So if we look at some of the data that we've generated recently using our polygenic risk score approach, we've been working extensively with the ADNI cohort, the Alzheimer's disease neuroimaging cohort, led by Professor Mike Weiner from UCLA. And in this case, we looked at 285 individuals who had early cognitive change, mild cognitive impairment, and asked whether or not we could have predicted those over a 4-year period who would declined significantly, and whether we could actually identify those at the level of genetic risk. And what we can see here with a 74% AUC, that the 61 cases who declined using the ADAS-Cog rating, clinical cognitive rating system, were at the highest end of the polygenic risk score distribution, whereas those who were still stable at the end of that 4-year period were at the lower end. So therefore, identifying and stratifying those using genetics only who are at highest risk of cognitive decline over a 4-year period, which of course is a period of a meaningful length for an individual patient, but also for a pharma study looking to show clinical benefit of their intervention. Now, if we look at the same data depicted in a slightly different way, on the left-hand graphic here, we can see that those who decline by the highest amount on the cognitive rating scale, the ADAS scale, 15 points are in red, or 5 to 15 in yellow. They're distributed at the right-hand end, the highest end of the polygenic risk score carriers, as one might have hoped. Conversely, those who remain stable or who decline by less than 5 points are distributed further to the left, at the lower end of the polygenic risk score range. If we look on the right-hand graphic, we look at the distribution by ApoE genotype. So we can see, as we would expect, that the E4 homozygotes are at the highest end of polygenic risk, whereas the E3, E4, so these would traditionally be considered to be equally high risk, are distributed, again, towards the right-hand end, but much more broadly across the range. So there are definitely some low-end E4 carriers within that profile. Very importantly as well, of course, are the common E3 homozygotes, who have a distribution from relatively low risk to very high risk. And of course, it's the high risk individuals that you would want to know about in terms of clinical management or in terms of recruitment into clinical trials if you're developing new drugs. So in conclusion, the stratification of the E3, E4 subjects can identify the highest subset of E4 carriers. And of course, these are the ones that would need intervention or inclusion into a clinical trial. The E3 homozygotes clearly include a group who can be shown to be at high genetic risk for progression and subsequent development of Alzheimer's disease, and that's very important clinically. The algorithm has the potential to predict individuals who will convert from MCI to AD over and above ApoE alone, as we've shown. And the GeneScore algorithm therefore has the potential to select MCI patients who are most likely to decline within the time frame of a clinical trial. That 4-year time frame is very important because that's the time within which pharma companies are looking to show statistically powered differences between control and treatment groups. And of course, you need a sufficient number of patients to decline within that period of the trial. Now, our GeneScore product is available for research use only at the moment. We have other algorithms that can also be run, for example, to examine the pathway SNP burden, the genetic variants on a pathway basis, which we believe have applications in identifying responders and non-responders to particular therapeutic interventions. And we can talk more about those offline if anyone's interested. And it's important also to say that we are seeking US and EU regulatory approval for GeneScore by the end of this year. And that's important because we know that Biogen have a very promising drug candidate in the form of aducanumab, an anti-amyloid monoclonal antibody that has been in discussion with the FDA since October last year. The FDA statisticians have been working with Biogen to analyze the data, and Biogen are planning to resubmit the full package later this year. with the expectation of an expedited review and at least some limited approval within the next year to 18 months. So it's important that a test such as ours is available to help clinicians identify and stratify patients for treatment once that drug becomes approved. I'd like to finish by acknowledging our collaborators. So we've got the Cytox team who've been working to develop this GenaScore test over the last few years and are behind the analysis that we've shown today. Our scientific partners, the key partners at Cardiff University, include Valentina Escott-Price and Julie Williams, who developed the original forms of these algorithms. And we work closely with Dementia Research Institute colleagues in the UK, Professor John Hardy at UCL, and Professor Clive Ballard at University of Exeter. And of course, the ADNI team in the US have been critical in supplying the data for these longitudinal studies. And Thermo Fisher are our partners on their instrumentation and the Axiom Array platform side. Finally, our funding, we'd like to acknowledge Innovate UK. We've had 2 grants from them, and that's been very important for the company. And we've had some funding as well from the European Union. I'd be delighted to answer any questions or have further discussion if you're interested. And if you'd like to get in touch with me, my contact details are on the screen now. Thanks very much for your attention.
Warum macht er nichts? Keiner redet.
Okay, and thank you very much again, Richard, also for your presentation. We do now have a couple of questions for you. Let me start with the first one, which is around actually the geography where you are already using GenaScore in fighting Alzheimer's disease. The question actually comes from Luxembourg. Can you tell us a little bit more about that?
Sure. Yeah. Thank you for the question. We've run successful studies in the US from multiple centres, in Europe, including the UK, and also in Australia so far. I think it's important to point out that the work we've done so far is limited really to Caucasian subjects of European origin. And that's simply a function of the fact that the genome-wide association data that you need to power these algorithms and derive the effect sizes of the SNPs is really not very available outside those particular ethnic groups, but we are keen to develop those in future. But essentially US, Europe, and Australia is where we've done most of our work so far. Okay, thank you very much for that. The second question for you, which is about, I mean, you have done this already partially in your talk, but again, I'm going to pose a question anyway, which is asking you if you can describe what are the key content that you designed onto the array and how was this important in order to generate the algorithm for a PRS? Sure. So the content of the array is really primarily informed by the DNA variants, the SNPs that are needed optimally to drive the polygenic risk score algorithm, which, and the commercial version of that we call GenaScore. We've got other algorithms on there as well. So GenaScore accounts for about 114,000 SNPs on the array. The array has 500,000 altogether. There are other algorithms that we can use this array to drive, and those SNPs are also represented on the array. And of course, we have a full suite of ApoE variants and also variants associated with early onset forms of Alzheimer's disease. These are much rarer, but incredibly important. So if we find these in a research sample, we flag these to the to the owner, the originator of those samples. And finally, we've got a lot of non-Alzheimer dementia content on the array as well. Not because we have polygenic risk scores, but again, it's important from a QC standpoint to know that the sample you're dealing with really hasn't, it's not an individual afflicted by different non-Alzheimer dementia.
Absolutely. So let me double check in case there's any additional questions from the audience. What do I see in here? So the other question would be, how has been your polygenic risk score validated? Again, you partially answered to this already in your presentation. So was there any independent verification, about that?
Absolutely. I mean, this ADNI data is independent validation. I mean, the original training of the algorithm was done in a different data set, an independent data set by the Cardiff group. We took the parameters from that algorithm, turned them into something that could be developed commercially, and then the validation in ADNI was completely independent. But we are working with a whole range of other data sets at the moment, longitudinal cohort studies. So because of the time it takes to develop Alzheimer's disease, what we tend to do is work with longitudinal cohorts that have been under study for several years, where a large proportion of those people have already progressed from maybe cognitively normal to early cognitive decline, or perhaps MCI to full AD, so we can test without having to wait for 5-10 years to see whether we were correct or not. So we've done a lot of that kind of work, and that's the sort of work that's going into our regulatory submission documents.
Thank you very much for that. And we actually have time only for one last question. Actually, it's a very concrete one, I'd say, and it's about how do you predict that the PRS would be used in the pharma space?
Okay, so the big challenge for pharma companies is not only to find subjects at high risk of developing Alzheimer's disease, but at the highest risk of cognitive decline within the follow-up period of that trial, because these trials are powered generally using cognitive test outcomes as well as other biomarker outcomes, and so you need to identify those people most at risk of significant cognitive decline using those testing batteries within a, say, 3 to 4 year period. So I think that's really where this polygenic risk score will find a huge benefit for not only for pharma companies, but also clinicians and their patients, because obviously these are the individuals who need closest clinical management.
Absolutely. Thank you very much for that, Richard. Actually, meanwhile, there's another last question for the first presentation, Jose, and that's actually about the turnaround time for prenatal reporting. So was there any difference between, turnaround time for the recipe, a known mutation, provided with the previous set of the tests, that you've done?
Yes, sure. So I have to say that everything was done retrospectively. So nothing was used actually to generate a report to send in to the medical doctor. But the potential is there that it will actually decrease a lot the turnaround time. So with the system, we can have it like in 2 days, 48 hours, we could have from sample to report being sent out to the medical doctor with the GenXs, whereas before it would take at least 5 working days to have it done.
Okay, thank you very much also for answering this question. So once again, thank you all very much for your presentation.